Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, in this lecture we are going to study the more details about the defensive mechanism of our body. Before discussing the details, I would like to say a few words about the previous lecture in which I introduce the terminology of immune system. Mainly it is of two types: innate, natural, or adaptive or acquired immunity. Adaptive immunity is again further subdivided into cell-mediated immune response and humoral immune response. Both of these types of the immunity, they are carried out with the help of T lymphocytes and B lymphocytes. Through this animation, I want to show you how antigen-antibody reaction take place, and as the result of this reaction, the lysis of the cell take place. You can see this cell to which the antibodies are attached. Whenever they come in contact with the antigen, they release specific substances. You can see that this is the antigen antibody reaction. Substances, they are released by these cells, B lymphocytes, because these are the B lymphocytes which secretes antibodies, and as a result you can see the lysis of the cell take place. So today's topic is how these antibodies they are produced and how this antigen antibody reaction take place, which are different steps involved in this reaction. For the formation of the antibodies, mainly the macrophages present antigen to nearby B cells, which afterwards enlarge and converted into lymphoblast, which changes to the plasmablast, and these are the plasma cells which produce antibodies. Mainly that happens, but few of the B cells they remain as the original clone and retain there as the memory cells. These cells circulate in the body and reaches to the lymphoid tissues for the production of secondary response. At this stage, I must tell you what is the primary response and what is the secondary response. Primary response is delayed, less intense and relatively short-lived, while the secondary response is immediate, more intense and lasts longer. What is the primary response? It is the first exposure of the body to the specific antigen. For that, the body is going to produce the antibodies. This process is time consuming, so the primary response is delayed. In nature, it is less intense and relatively short-lived, while the secondary response is the second response, It is immediate because this time we have already some of the antibodies there. Because of that, the reaction is more intense and it lasts longer, maybe for the whole of the life. This is the summary how the macrophage present antigen to the B cells, which changes to the lymphoblast, changes to the plasmablasts, and in the end these are the plasma cells which are going to produce the antibodies. This is the main reaction or changes, while some of these cells they remain as such, circulate and come to the lymphoid tissue for the later on response. And because of this we have primary response and followed by the secondary response. Through this graph I want to show you that how this primary and secondary responses occur. This is the first exposure of the antigen to our body and it is delayed. As I have already told you that for the antigen, antibodies must be produced by our defensive cells. It is time consuming, so the reaction is delayed. Primary response is there after the first exposure. Then with the time it subsides. But some of the original cells they remain there to have a good enhanced response later on. So, if ever we have the antigen exposure for the second time, our antibodies are there in our body so that at this time there is no delay. The response of the body is relatively immediate as compared to the primary response. And as memory cells are there, they remember what happens in the past. So the response at this time is much more enhanced, as you can see here, and it lasts longer, maybe for the whole of the life. Compare this one to this and compare the peaks of primary and the secondary responses. There is a great difference between these. Now, the nature of the antibodies, what are actually these one? The antibodies are actually the gamma globulins with high molecular weight. Structurally, they consist of two identical light and two identical heavy chains held together by disulfide bonds. At the end of the light and heavy chains, we have the variable portion which is named as Fab portion. This is the portion which is different for each antibody and attaches to the antigen. And this is the portion which is different because and different and specific for each specific antigen. As the antigen changes, so that this portion also changes accordingly. While the other portion, that is the remaining portion, is fixed or constant and called Fc portion. And at this, the adherence to the structure attachment to the complement protein take place. So at this end, the antigen is going to be attached and at this attachment of the complement proteins take place. A very nice picture to show you the two identical light chains and two identical heavy chains. This is the upper portion for the attachment of the antigen, which is variable depending on different type of the antigen. Why it is variable? Because we have variable or different types of the antigen. So it is very much specific for each specific type of the antigen. While this remaining portion, that is this one, it is constant and it is called Fc portion. These chains, they are interconnected or joined together by disulfide linkages, while this portion, that is the arm or hinges, they are flexible for the good and strong attachment of antigen to the antibodies. All immunoglobulins are not antibodies. Mainly we have 5 kinds of the antibodies, and in short they are called as simply game D. This is the picture for each and every type of the immunoglobulin. Remember this word, that is game D. I write here as, from this word you can easily remember the types of the main immunoglobulins. Among that, the most abundant is IgG, which is 70 to 75% of the total immunoglobulins. At this stage, I must tell you that these things, they are not written in your textbook. So be careful about these slides, and especially the very specific characteristic of these I have already highlighted, that IgG is the specific immunoglobulin which can cross the placenta. Coming to IgM, it is secreted initially during primary infection and it cannot cross the placenta. IgA, it is in the dimeric with secretory component in the lumen of the GIT and in the respiratory tract, and it neutralizes microbes and toxins. IgD present on the surface of B lymphocyte and it functions as membrane receptor. Its exact role is not clear and has a maybe having a role in antigen stimulated lymphocyte differentiation. Coming to IgE, you must remember that they are very important regarding the hypersensitivity reaction. I will tell you the hypersensitivity reaction details of that later on, but I introduced this hypersensitivity reaction terminology in previous lecture also while discussing the eosinophils and basophils. Later on, we will discuss the hypersensitivity reaction, allergic reaction in much more detail. But at this stage, you must remember that which type of the immunoglobulin they are important for hypersensitivity reaction. The answer is IgE. About IgM and IgG, we have a very sequential humoral response, very specific response, as IgM produce is the first response to many antigens and IgG produced after IgM. Through this very nice graph, I want to show you that with the first stimulus, the IgM, they are released. But they remain there, while with the second response, we can have IgG also. Now, coming to the next thing, that is, what is the main function of the antibodies? As we know that after the production of antibodies, this production is in response to the antigen. So, antigen antibody reaction take place, and as a result, the cell or antigen is killed. This reaction or this reaction may be of different type. There may be agglutination, precipitation, neutralization or lysis of the cell take place. In the process of agglutination, multiple bacteria or RBC are bound into clump formation. They settle down as one over the other, forming the clumping of these cells. In the process of precipitation, soluble toxins, antigen after combining with antibodies are made insoluble and the precipitate that laid down. Neutralization, in that process, antibodies cover toxic sites of antigens like viruses and toxins. While in lysis, breakdown take place, antibodies directly attack cell membrane and cause its rupture. Through this figure, I want to show you how all these mechanisms take place. In this, you can see the antigen, maybe bacteria or virus. They are attached or attacked by the antibodies. So the antigen antibody reaction take place, and this is the mechanism through the neutralization, agglutination, that is, they are placed one over the other, then precipitation, activation of the complement system, which will be discussed in the following slides, and then we have the cell lysis either through this process or through the phagocytosis. The end result is the death of the infective agent. Indirectly, we have the following action, mainly causing amplification of the innate response. There may be activation of the complement system. What is that one? That will be discussed next. Enhancing phagocytosis, as I have shown in previous figure, and stimulating the natural killer cells. What are these one? These cells have receptors for constant portion which link target cell to the natural killer cells. Now coming the main topic for the defensive mechanism of our body, we have a great role played by the complement system. What is this one? More than 20 proteins produced by the liver and present in inactive form in blood and tissues. Named from C1 through C9, B and D. What are these? They are different 20 types of the different proteins. Again, the main action is to defend our body. For that one, we have the complement system, which is actually the combination of three basic pathways: the classical pathway, alternate pathway, and the lectin pathway. When you will go to the high classes in third year, in fourth year, you will study the much more detail of this complement system. It is not so simple as I am telling you at this stage. It is a very complex system running many different mechanisms side by side. So it is not so simple as it looks from one figure, which I will show you in the next slide. It is the combination of many pathways and it involves many steps. How it is activated? It is activated by the antigen, antibody reaction. I have told you previously that whenever our body is attacked by any type of antigen, our body mechanisms, they start, they come in contact with these antigens. The antigen antibody reaction or our defensive mechanism whole story depend upon the number of the antigen, its severity, damage to the tissues, how much, how widely spread the antigens they are. All these factors they cause the extension of these defensive mechanism. If it is very simple antigen, very simple type, very few antigen are there, very less injury to the tissue. So the first line and second line, natural second line phagocytic cells like monocyte, macrophage, neutrophil, they are sufficient and the story stops there, finishes. But if they are not killed completely, other complex mechanisms they also play great role to control that infection. But it doesn't mean that the role of neutrophil macrophages that stops. They are doing their job, but other more complex systems also play their role. Among these complex mechanisms, we have the complement system, which is mainly the combination of three basic pathways, that is the classical pathway, alternate pathway, and the lectin pathway. Classical pathway, after antigen binds with antibody, a site on Fc portion becomes naked and C1 attaches to it. It activates C4 and C2 and active part of C4 and C2 activates C3 and form C3A and C3B. Products help to prevent damage to the tissues. What is the alternate pathway? It accounts for the majority of terminal pathway activation. So initially we have the classical pathway. Then with that alternate pathway also assisting this main complement system. The alternate pathway is continuously activated at a low level as a result of spontaneous C3 hydrolysis due to the breakdown of internal thiazide bond. Now, what is the lectin pathway, which is the third important pathway? It is homologous to the classical pathway but with some difference. Instead of C1q, this pathway is activated by binding of MBL2 mannose residue on the pathogen surface, which activates MBL assisted serine proteases, which can then split C4 into C4a and C4b, C2 into C2a and C2b, C4b and C2b, then bind together to form the classical 3,3 convertase as in the classical pathway. So at this stage, they join the main classical pathway. This is the main complement system. You can see the antigen antibody complex, antigen antibody reaction take place here, which initiate a number of steps so that in the end the lysis of cell take place. This is the main pathway, but other pathways, for example alternative pathway or other pathways, lectin pathway entering here, they are assisting the main chain, and not only the end product is this one, but at different steps we have this also. That is, the mast cells and basophil, they are activated, they come to play more and more. Mast cell and basophil come to play their role. Opsonization of bacteria take place, chemotaxis of the white blood cells at this step. So it is not so simple that these steps take. And I am showing a very simplest figure regarding the complement system. When you go in higher classes, you will recognize that this is not so simple. It has many other steps and it is very complex system. At any stage, if we succeed to kill the infective organism completely, all these mechanism stops there. They will not further go ahead. So, if we become successful at this stage, that is the steps following the main line, if they assist and become successful to kill completely the bacteria, then no need to have further steps or initiation of more mechanisms. At this stage, I must tell you what is opsonization and how the lysis take place. Opsonization, that is C3p activates phagocytosis by neutrophil macrophages by attaching to the bacteria. C5b6789 forms membrane attack complex. Agglutination, that is the product chain surface of bacteria which adhere to each other. Neutralization, chemotaxis, activation of mast cell and basophil, as I have shown all these things in the figure, and we had the inflammatory effects, directly increased blood flow, capillary leakage and coagulation in tissues, and much more steps and processes regarding this main complement system. So in this way, our body defense occur against the infective agents. We will discuss these processes in further detail in following lectures. So keep with me and keep watching these lectures carefully. Okay, thanks and Allah Hafiz.